It's a pleasure really to be presenting here some of the work that we've been doing. This is really emergent findings, but at the same time sort of gives you an idea of some of the work that we're trying to examine in the next few years.
Could you speak up a bit more, actually?
This is the work that we're trying to do in the next few years based on this really rich opportunity to study a database which is being created right here in Montreal based at Douglas Hospital project I'm going to talk about. So I'm going to talk a little bit about anxiety and depressive disorders in children, what we know about the roles of various etiological factors like prenatal experience, genes, and then early postnatal experience. I'm going to talk about how it's important really to research a more complex model to understand the causes if we're going to be investing in early intervention and prevention in ways that are meaningful. And I'm going to present some of the early findings from a project which aims to do that exactly, and as well what are the implications for clinical practice. So anxiety and depression in children affects children really as early as in the preschool age period. Surprisingly, you can actually measure anxiety in children starting at the age of 2 to 5, and it's almost the same prevalence as in children who are 6 to 18. It rises when you enter adulthood, and when it rises to 18%. Depression is a little bit less prevalent, 2% in the preschool age period. It rises to 4%. I'm getting a lot of feedback, I'm sorry. In the 6 to 18 year age period, and then after that rises to 7% later on. What is significant is really that some of this pathology continues into adulthood, and it has impairments in school, in peer relationships, and in family functioning, even in the preschool age and early elementary age period. And even more significant is that 50% of children with psychiatric disorders during the school age period can be identified using parent reports of symptoms at the age of 2 to 3. So there's a continuity. It's been considered the leading health problem that Canadian children face after infancy. There's evidence for the effectiveness of early treatment of anxiety disorders in elementary school age children and preschool children, but there's less evidence about prevention. There's some elementary school-based interventions. There's no evidence really for, almost none, for preschool children for intervening in early enough before they even have the onset of any symptoms. There's been one parent-based intervention for children of inhibited preschool children that found an impact at 12 months and at 3 years of age. This is based in Australia. But even then, if you read the fine lines, some of these children were actually exhibiting some of the first symptoms, and with some of the associated incapacities, you might even make an argument that some of them already had some disorder. But what I'm going to argue is really that for prevention and early intervention to exploit the capacity for neuroplasticity in early childhood, the fact that the brain is developing and only about 0.25% of it is formed in its final state at the time of birth, that we need to have a better understanding of causal and protective factors in the trajectory for young children, as well as early markers. Something that we can say, well, maybe this is a child that can, we can target, refine our target children. And then findings associating prenatal experience with anxious and depressive psychology provides interesting directions. So certainly there's been a lot more airtime recently about the importance of the prenatal period. It had been understood to be a bit of a cocoon that was protecting the children from every experience that the mother was having. There are a few people who had talked and delved about the importance of prenatal experience who certainly didn't seem to get the same scientific airtime. I'm thinking of people like Arthur Janov and Primal Scream and the experience of prenatal experiences being, leading down roots and leading down experiences that are scarring that had to be undone in therapy after one was born. Certainly there's been a resurgence of the research in this literature, and I'm going to talk a little bit about why there is actually very good convincing evidence that that period is not as unimportant in the development of an organism, and that when one is born at 9 months of age, conceptually that there's actually been 9 months of age of experience. So in 1989, Barker developed something called a hypothesis, which basically said that there are a lot of diseases that originate from undernutrition during fetal life. He actually now on his website, it's no longer called a hypothesis, it's called a theorem. He believes that there's enough evidence that now we can call it a theory so that it's well-grounded. And that in response really to changes in the intrauterine environment, Gluckman and Hansen said, such as nutritional restrictions or stress, that the fetus will make adaptations to improve the chances of survival. So there's always a constant dialogue between the outside world and the inside world. And that the fetus in developing adapts its phenotype, meaning it's the way it's going to look like, as a means of adapting to the conditions of the anticipated postnatal environment, that there's something happening, signals that are being told about what to expect in the environment, and one has to actually develop accordingly in order to prepare ones for increasing the chances of survival. So we certainly know that that prenatal anxiety and depression, especially characterized by catastrophic events, prenatal life events, and even chronic social stressors have been important in terms of the impact on birth weight and preterm labor. There's also been some important evidence on how it's associated with temperament, such as fussiness, negative behavioral reactivity, and fearful temperament. I'm hearing a bit of feedback. I'm not sure everybody else is.
Yes, definitely.
Okay. I'm not sure what I can do about it. Is this one work?
It does, but if you push it away, maybe we
Move it away.
How about this? I mean, I'm happy to turn this off and let's speak out of the other microphone. How's this? No. Totally turned off now. Is it on now? Yes. So it's interesting. Okay. I'm not sure. Okay. So, and there's also been, it's also associated with cognitive and motor developments, such as inattention and processing speed and spatial learning. And it's also been associated with social and emotional development, such as anxiety, depression, and conduct disorder. That part, the anxiety and the depression, is a lot less well documented, and this has really been the target of the research that I've been endeavoring in. So, of course, we know that it's not all about the prenatal experience, that genes certainly have an important contribution. There's certainly a time where we thought genes was the significant contribution that was going to help us explain what was the psychopathology of children. And one of the big genes that have had a lot of airtime, especially in development of anxious and depressive psychopathology, is the serotonin transporter gene. That's really been a gene that's integral in the transcription of serotonin and in the synapses and the activity of serotonergic nervous system. And the gene is organized in a way that there are variations in it, that some people might have. We always have two versions of a gene, what we call alleles. It's a short and a long form, and depending on which combination you have, then you might have more or less transcription of related to the serotonergic system. And certainly this gene has been associated with psychopathology, where when you have two short alleles, that's been associated with predispositions to anxiety and negative emotionality. However, the data for children has been a lot more inconsistent, and there's been a lot of debate as well as exactly how to measure the activity of that gene. So, but at the same time, I think as time evolved, especially as the Genome Project really finished its objectives, people thought, well, maybe there's more to genes than we thought. We can't really explain most of the conditions based on one single gene, and maybe there's something else about genes that we don't understand. And one of the hints to understand that is the fact that if we compare humans and worm, we actually have only 50% more genes than the worm does. But actually, the size of our genome is 30 times larger. And this was why initially we thought we would have 200,000 genes, 300,000 genes, and slowly we kept refining that down until we now have only identified 30,000 genes. And the reason is the following. And you can see here what is green are called the gene coding regions, and what are white are what originally thought to be junk regions, areas that were meaningless. And you can see that in the humans there are a lot of white regions. And these regions here are basically regions that aren't specifically coding, but they're regions that are related to the coding regions by regulating whether they actually are active or not. I mean, this is as technical as I'm going to get in this talk, but the important part to retain from all of this is the fact that we start to realize that genes just don't code automatically. There's some that code all the time, there's some that actually code some of the time, and there's some that code none of the time, and there are periods in life that actually influence the activity of those genes. And so we started revising understanding of whether these areas were meaningless or not, and realized that there are regions called promoter regions or silencing regions that actually make an enormous difference as to whether a gene is actually active or inactive. And to the point where a famous McGill professor, Donald Hebb, said, to ask which contributes more to development of personality, nature, or nurture is really like asking what contributes more to the area of a rectangle, the length or the width. The genes really operate in an environment, and that environment has an impact on whether the genes are active or not. So obviously, we thought, well, there are obvious postnatal experiences that are meaningful. We've had a lot of evidence about the role of trauma, the role of catastrophes, the role of stressors. But I think the project is born out of the idea that maybe there are more sort of lower degrees of exposures that are more important and more prevalent that also make an important difference in the development of children, such as parental care. And this board from Ellen Moss who presented earlier this year, last year, in terms of the role of attachment, and showed that certainly there's been very good animal research at rats showing that the type of behavior that rats have with their pups can actually influence the outcome of their offsprings. And actually this experimental research has been really elaborated at length in very meticulous and beautiful fashion by Dr. Michael Meany, who, as you've heard, is the actual director of this project, which really showed that the type of maternal experience that children had earlier on, and actually whether or not they were biological offsprings of that rat mother or whether they had been adopted early on, actually had an influence on their stress response, had an influence on their learning experience, had an influence even on how their brain looked in terms of the number of nerves that had developed inside the brains, and had an impact even on the genes and the activities of those genes. So, of course, we don't lick and sort of nurse our offsprings as often as rats do, but there are equivalent sensitivity or equivalent maternal behaviors that we think would be targets of research if we wanted to understand, well, what exactly in the early maternal child experience could we say makes a difference in the evolution of children. And so we decided to focus more on sensitivity, which you're going to hear a lot more about, but really is a coordination of interaction, the reciprocal and rhythmic gaze, cycles of behaviors in different modalities, so gaze, touch, affective expression, body-oriented. This is attunement between a mother and a child. And that's been associated in studies already with behavioral changes in the children, that optimal maternal sensitivity has been shown to be associated with decreased behavioral changes, whereas intrusiveness, overprotection, overcontrol are associated with behavioral problems. And even more interestingly is that children who are high risk, like children who are low birth weight, for example, have the postnatal experience, the experience with their parent, as measured in usually very meticulous methods, shows that, for example, with low birth weight children, that low birth weight children with more sensitive maternal care have less anxious and depressive psychology at age 8. Or that mothers who have more reciprocal play and have better maternal play quality at 8 months improve the mental development at 18 months of low birth weight children with fussy temperament. So we know that it actually makes a difference, and it can make a difference even more for some children who have some particular risk. In prenatal anxiety, there's really very little emerging literature on this. There's one study that was presented last year that basically showed that optimal maternal care behavior reduces the effect of prenatal maternal stress on cognitive development. So, and the idea is prenatal adversity always bad? I mean, I think that's the last point that I want to highlight before I actually enter, and hence the title of this talk. Because our principal understanding of mental disorders is that stress interacts with vulnerabilities to lead to poor adaptation and manifestations of psychopathology. And Belsky and Pless refined this observation and said, well, maybe some organisms are more affected by positive or negative environments than others, what he called for better and for worse, an idea called differential susceptibility. so that the experience of some prenatal experiences, or maybe even some genes, make or children at higher, more attuned to what happens in your life when you're born. So that if you have positive experiences, you might actually have a better outcome having these what were initially called risk genes, that they might actually be susceptibility genes. So some children exposed to prenatal adversity and with risk genes might actually fare better when exposed to an enriched environment. And the operating sort of concept is what's happening after you're born. And in a way, this is most of what we're all involved in is the postnatal period. But I appreciate that our audience is constituted people who are also working in perinatal care, because we, looking at your targets of intervention that span the whole developmental spectrum of children. So our model really is that prenatal risk, characterized by prenatal maternal depression, and genetic risk, in this case characterized by serotonin transporter, will be influenced by the postnatal environment to predict anxious and depressive symptoms. And of course, this is adjusting for other variables that are other factors that we know explain psychopathology, like the sex of the child, social economic stresses, maternal depression, postnatally, even life stressors. So the project then that I've been involved in, I've been very fortunate to be involved in the psychiatric outcomes, is the MAVEN. And really the MAVEN is a project that had the following hypothesis: the differential effects of maternal risk factors during pregnancy on the development and behavior of the child are closely associated with the genes and also the quality of the environment. So the mental health objectives are: what are the developmental risk factors for early age anxious and depressive psychopathology? How does the early environment modify the risk for development of early and anxious depressive psychopathology? Are there early markers that we can actually then refine the targets of intervention, such as temperamental characteristics? And does prenatal adversity always lead to negative outcome? So the MAVEN project was constituted from mothers who were recruited during the prenatal period in university hospitals at McGill, at University of Montreal, and at McMaster, so in Montreal and Hamilton. In Montreal, they were oversampled for mothers with risk for intrauterine growth retardation, so lower birth weight. Versus in Hamilton, they were oversampled, meaning they tried to get more mothers who had depressive, anxious experiences during the pregnancy. And in total, that made for a sample of about 600 children. All about 18, the mothers had to be 18 years of age at the expected date of delivery. It was a singleton pregnancy, and they had to be either fluent in English or French, the languages in which all the questionnaires and the laboratory measures are administered. There are hundreds and hundreds of laboratory measures that have been administered to these mothers over the period. Now the oldest is about 9 years of age. I'm going to present a few of them. But keep in mind that there are other outcomes that people are looking at, such as cardiovascular, metabolic, sleep, cognitive, attachment. These are, because all of these have been certainly thought to be related to prenatal experience. But the exclusion criteria were that the mothers could not have severe chronic illness requiring treatment or obstetrical pathologies, there couldn't be major congenital anomalies or premature birth, and there couldn't be significant developmental delays. And prenatal adversity was measured using a scale called the Center for Epidemiologic Studies Depression Scale at trimester 3. So mothers were asked to complete a questionnaire that had 20 items about some of the symptoms that they were experiencing during the pregnancy. And this has been validated as well as in pregnant women. As well, we certainly all the genes are being looked at, and other more refined aspects of the genes, but just to say that at least sort of the serotonin transporter gene is one of the ones that has, that is being assessed. And as I was speaking about the actual postnatal experiences being examined for one, looking at maternal care across the developing spectrum. I'm only presenting it at 6 months of age, but it's being examined all the way up to 5 years of age. So the mother-infant dyad participates in a 20-minute videotaped non-feeding interaction, followed by 5-minute divided attention tasks. So in total, there's 25 minutes, and mothers are asked to interact normally with their child. And the mothers are rated in their interaction with an inter-observer reliability about 70 to 80 to 100%. And they're rated on numerous different, like second-by-second measurements of how often they look at their child, how often they look away from the child, whether there's passive attention, joint focus, whether they're touching, kissing, stroking, patting, singing, stroking, rocking the child, jiggling. So there are various different dimensions. I'm going to speak today just about the one that has to do with maternal attention, and looking away. The idea is that if the idea of maternal sensitivity is being attuned to the clues and the cues that the child is giving about their needs, and if you are looking away from the child, not 1 or 2 times, but 50 times, 70 times, or for the majority of the duration of the time that you are with your child, then it is really hard to get all the information you need to know about what's happening with your child. So it is really inversely related to maternal sensitivity. And the outcomes are numerous, but I'm presenting the ones that has to do with temperament. So maybe it's something already at birth, being as close to birth as possible, being signaled that that the child has actually responded to experiences during the pregnancy, and also at 2 years of age. So at birth, the temperament is the infant behavior questionnaire that we extracted a factor called negative emotionality. It's sadness, fearfulness, avoidance. Versus, and at 2 years of age, we use a child psychopathology, a first measure that we have called the ITSE, the Infant Toddler Social Emotional Assessment. It looks at externalizing and internalizing symptoms. It also looks at dysregulation and competence, but I'm presenting symptoms related to internalizing, so withdrawal and depression. So, just to give you an idea for the type of sample that we have, you can see here that there's a full range of ages, and some of the children are still under 12 months of age, while some of them are over like 6 years and 8 years of age. It was an equal distribution of boys and girls. The average birth weight was about 3,300 grams, with an equal distribution across the different quartiles of weight distribution, and about 14% were under the 10th percentile. And that's, I think reflects our sampling strategy because it's usually about 10% by definition. And there's very little developmental concerns about these children. In terms of the genotype, there was a distribution. Both of them had the long alleles of the serotonin transporter. 36% had a heterozygous, a long and a short, and then some of them had just the short. As well as the mothers, you can see they're 30 years of age, 95% were married or had at least there were 2 parents at the time of the birth of the child. You can see that half of them at least had some university education, and half of them at least had $60,000 of income. So we certainly have, very positively favored sample. 30% met the cutoff for depression according to the CSD, and the average score was 12.8. And then the below one, the last item here looks at the stressors, how many hassles. On average, it was like 1 hassle. Hassle meaning like, poverty, missing money, or having to move, or something, or somebody sick in the family, versus trauma, which is like assault, a rape, attack with a weapon. That was 0.1. So there is almost a very, very rare occurrence of traumatic events. Just to give you an idea of the distribution of the score of depressive, this is, like I said, the CSD gives 20 items. You can have a score. The 16 is the cutoff for depression according to the CSD, and you can see that the majority, of course, of the samples, both in Hamilton and Montreal, have below cutoff. But there's a nice distribution across the full range of the measure. And this gives you an idea of the type of frequency that mothers are looking at away from their children in a 25-minute period. And you can see that there's not necessarily a normal distribution. There's maybe it tails a little bit more to the right, but around 20 times in a 25-minute period is the average that mothers might be looking away from their child. But there are some mothers who are looking away 39 times, 45 times, 70 times. And what is important is looking away is not necessarily a negative aspect of an interaction, because sometimes you need to look away in order to help the child re-regulate themselves if they're overstimulated. But doing it 70 times and spend 25 minutes, and the duration statistics are even more significant, because then you start to see 15 or 16 minutes of the time that the mother was with the child, they actually weren't looking at them. So in terms of negative emotionalities, this is just to give you an idea that the way we created the factor, it's actually quite nicely distributed from people with children with more negative emotionality and some with less negative emotionality. This is one of a couple technical slides I'm going to present. Really what this is saying is, if we're trying to predict temperament, at 3 months of age, if we're trying to predict temperament at 3 months of age, then not only are genes significant, but also genes and prenatal depression. And I'm going to show you a graph to show you that. Basically, the ones in red are the children who have only the long allele. The ones in blue have one of the short alleles, and the shorter allele is considered to be the risk allele. And what is this showing you is that if you have that risk gene, the number of symptoms that your mother reports of depression during the pregnancy, as the number of symptoms the mother reports during the pregnancy increases, those with the short allele, which is a risk allele, have more negative emotionality already characterized at 3 months of age. So it's already saying that that it actually matters what was the prenatal experiences as well as what are the genes in terms of predicting temperament. And in terms of anxious symptoms at the age of 2, I'm going to present this other graph instead of presenting the data. The data just, the tables are just showing you that actually these analyses are all adjusted for sex, income, and for even maternal depression at the time of the study of the intake. But this is showing you if the mother did not have any depression during the pregnancy and the infant had the long alleles, then regardless of what type of depressive symptoms the mother had, really they had almost no depressive symptoms, the child. But if the child actually had the short allele, either one or two of them, as the number of prenatal depressive symptoms increase in the mother, so does the number of symptoms that the child has at the age of 2. So it's saying that it really matters not only whether the mother was depressed, but it also matters what type of genes you have and also what type of experience that the mother offers. So this is, as the mother looks more and more away from the child, these children have more and more depressed symptoms. And this is what happens if the mother actually had depression, according to the cutoff. You can still see that if the mother looks more away from the child, that the number of symptoms increases. And if the child didn't have any of the risk genes, then there's almost no difference how many times it looks away or doesn't look away. What I would like to highlight is that this part of the graph really shows you, is it possible that even if you have the risk gene, so if you're in the blue, the serotonin transporter short allele, is it possible that if you have a good enriching environment that has a very sensitive experience, you might actually have fewer symptoms than those who didn't have the risk genes? Because here you can see that it's almost, the line on the left is even lower than those without the risk gene. So this we actually have to study more precisely, but the idea is maybe it's not deterministic, the type of genes that you have, and maybe it's not deterministic, the type of prenatal experience that you have. So just comparing those two graphs. These are modest changes. We're looking at measures that sometimes are reported by the parents. We only use one measure of maternal care. We've only used one gene. We haven't adjusted yet for life stressors. And there's some questions about the optimal coding of the serotonin transporter. Really, why this is important in terms of intervention is, first of all, that prenatal maternal depression is formally diagnosed in 9% of pregnant women and experienced in some degrees in terms of symptoms in 50% during pregnancy. That postnatal maternal care is associated, there's evidence that one can modify maternal care, maternal experience, that there's evidence about factors which modify the development of early anxious depressive psychopathology, can inform development of parent-child intervention. So there are good studies that show that if you actually modify sensitivity in ways that, for example, Ellen Moss showed us last year when she came to present here, you can actually have an important impact on the behavior of the parents, so that is actually modifiable. And finally, that temperament as an early signal is something that we might even consider to be modifiable. These early signs of child psychopathology might be an early marker of children who are susceptible, and if we intervene early enough, these markers might tell us which children would be best for us to target our intervention. And for example, that there's evidence already that we can modify such temperamental characteristics as behavioral inhibition. There's one study that found that negative emotionality moderates the relationship between early intervention outcome for children who are premature and low birth weight. So it's really saying that maybe these children who have fussy temperament might actually be more receptive to interventions. So there's evidence that these are actually points of intervention might be good for us to explore in the long run. So I just want to thank everybody who's been involved in this. I'm really a humble participant in this project. There have been numerous investigators and collaborators. Martin Santandré is actually a discussant here, but he's also been an integral part of developing this project. So thank you again for your time.